Ben, are you about ready to start?
Sure.
Okay, so I will start then. All right, so we'll start this presentation now. Um, Good morning. I'm Joe Liu from UCLA, and it's my pleasure to introduce our seminar speaker today. But before I do that, so Ben Wei is giving the presentation. He's currently at Merck in Kenilworth, New Jersey. But before I go through the formal introduction, let me just introduce you then to the consortium if you've not been to these meetings before. Oops. We want to continue to add our thanks to our sponsors for the consortium for top-down proteomics. That's Seer and all the companies that you see listed here. They make these kinds of presentations possible. So without their support, this doesn't happen. So please thank them if you see representatives from these companies. Ugh. Also, then we try to have these presentations once a month on a Thursday. We've invited a lot of great young people working in the field of mass spectrometry and top-down proteomics. If you know of anybody who would be an interesting speaker and could give a good presentation, please forward their names or your name to our fearless leader, Paul Danis, working in the office. The website is here, or just put a comment or a name in the chat as you listen to today's presentation. So we are always looking for new people who've not presented yet, who have some great work in the field of top-down proteomics or proteoform technology. Also, ASMS, the annual meeting is coming up in Anaheim. And so it's my great pleasure then to invite you to come to the activities. As Vice President for Programs for ASMS, I know a lot about the programs. We have evening workshops scheduled for Monday, Tuesday, and Wednesday of the conference. And on June 3rd, then the workshop on top-down proteomics we'll be giving, we'll have an evening workshop. The topic is bridging the gaps between academia, clinical research, and biopharmaceutical research. It's going to be organized by Fanny Liu from Florida State University and Yuri Vanderberg from Leiden University in the Netherlands. And you can see the topics that they'll be addressing. There'll be an update on the interlaboratory research projects addressing some of the technical challenges in top-down proteomics, and discussions on fundamental clinical research and research in the biopharmaceutical industry related to this particular topic. And so you're encouraged to attend. You're encouraged to join the discussion, and you're encouraged to propose new topics for discussion on this evening workshop. So please attend if you can during the annual ASMS conference. Also, if you're interested to enroll in the top-down proteomics short course that'll be held on the Saturday and Sunday before the conference starts, you're invited to enroll. I think there's still time to enroll, and there's still slots then for people to attend. It's a great this is my unbiased view since I'm one of the instructors, but it's an unbiased view of how good this workshop can be. If you're just getting into the field, it's a great place to start learning how to do top-down proteomics. So you can see the lists of topics that would be covered: history, sample prep, how to do protein separations, the actual how to do fragmentation of proteins, et cetera. And then on the second day, the second day is devoted exclusively to software tools, hands-on training on using some of the key top bioinformatic tools that's used for protein interpretation, protein identification and quantification. You can sign up for just the Saturday short course or the Sunday short course separately, or you can do them combined. So that's up to you. But please consider registering for the top-down short course. Okay, so with that, then let me introduce today's speaker. That's Ben Wei from Merck. Ben started his academic career. He received a bachelor's degree in engineering from Sichuan University in China in polymer materials and engineering. Then he moved to the United States. He moved to the University of Akron, where he received a master's of science degree in polymer science with specialization in mass spectrometry. And there, he worked with my friend Chris Westamiotis in the chemistry department at Akron. Um, I was fortunate enough to recruit Ben to UCLA, so he spent some time in my laboratory, and he received his PhD at UCLA, and he left UCLA at the end of 2023. Ben was a highly productive student in my laboratory. He published, oh, I forgot now, at least 8 publications. Half of them he was first author. He published a book chapter, and while he was at UCLA, he received many awards, including an ASMS Graduate Student Travel Award. At UCLA, he received one of our highest honors for research, the Roberts A. Smith Excellence in Research Award at UCLA. And he received the American Chemical Society Analytical Graduate Fellowship Award that was sponsored by Agilent in 2023. So while Ben was in my laboratory, we started a project on applying top-down and middle-down mass spectrometry to characterize therapeutic proteins. And today's talk, he's going to highlight a lot of that work with a special emphasis on the utility for internal fragments to obtain more sequence information. So with that very brief introduction, then I'll then allow Ben then to take it over then. So Ben, why don't you go ahead and start your presentation?
Let me share my screen.
Great, got it.
Oh, all right. Thank you. Thanks, Joe, for the kind introduction. I first of all, I want to thank Consortium for Top-Down Proteomics for giving me this opportunity to present my my graduate research work in Joe's lab. And as Joe introduced, I was a student in his lab for the last 4 to 5 years. I graduated at the end of last year, November last year, and then I moved to all the way from the West Coast, the warm, sunny West Coast to the East Coast, right now in New Jersey. So I joined Merck after graduating from Joe's lab. I had a great experience there, so thank you, Joe. So as Joe introduced, the topic of my presentation today is about using internal fragments in top-down and middle-down mass spectrometry. I'll start from fundamentals and then move on to applications. So this is a general outline of my presentation today. I will start by introducing what are internal fragments and why are they important. And then I will talk about 3 specific projects. The first one is about fundamentals to understand how internal fragments are formed. And then 2 applications of using internal fragments for top-down and middle-down characterization of biotherapeutic proteins. So first about internal fragments. As we all know that mass spectrometry is an important tool for protein analysis. And this is usually done by 3 different ways. The first and the most popular and traditional way is bottom-up, in which we digest our protein of interest into small peptides, which will be separated by chromatography, and then we do fragmentation to generate sequence informative fragments of these peptides. On the other hand, we also have top-down mass spectrometry, in which we do not do any digestion. We directly analyze our protein of interest. We then either do direct infusion or we still separate them by chromatography. We can then isolate, select and isolate the proteoform that we're interested in, and then do fragmentation. In the middle, a relatively new method called middle-down mass spectrometry In this case, we reduce our protein into large subunits, or we do limited digestion to large peptides. And these subunits or peptides will be again separated by chromatography, and then we do fragmentation. So all these three different methods have their own pros and cons, but a common theme of them is this step, the last step of fragmentation, which is also what I was very interested in during the graduate school. So traditionally, when we think about fragmentation, we only consider terminal fragments, which are generated by cleaving once on the protein backbone, right? But if we only consider terminal fragments, sometimes there's a limitation, which is that sometimes we cannot access the middle part of the sequence, so we might just miss the central protein sequence information. However, if we can also consider internal fragments, which are usually generated by cleaving twice or even multiple times along the protein backbone, there's a chance that we can piece both terminal and internal fragments together to get the full protein sequence information. So this is an example to show that both terminal and internal fragments actually exist in a top-down mass spectrometry experiment. So this is a ECD MSMS spectrum of NISMAP FD subunit. If we zoom into the low M/Z region, and we can actually see a lot of fragments, and we can assign them as both terminal and internal fragments. And a lot of the previously unassigned signals cannot be assigned as terminal fragments, can now actually be assigned as internal fragments. And a lot of these internal fragments are much lower in intensity compared to terminal fragments. So we usually do a validation to make sure that they are real ions. So I hope this example have shown you that both terminal and internal fragments exist in a top-down mass spectrometry experiment, and these internal fragments can provide additional information to complement terminal fragments. However, right now our field, our top-down field, is not really. I mean, it's largely ignored. The assignment, the analysis of internal fragments is largely ignored by our field, mainly because that there are too many theoretical possibilities of them compared to terminal fragments. So first, it requires a large computational demand to assign them, and also there's a high chance of false positive rate if we want to consider internal fragments. So to help, so the goal of my graduate school research was to kind of to promote the application of internal fragments. So to do that, we have to understand them better so that we have higher confidence to assign them with higher accuracy. So that's why we started by doing a fundamental study to try to understand the formation of internal fragments. So this is just a very simple peptide fragmentation scheme. If we do a CAD, or collisionally activated dissociation, it will cleave the peptide bond here to generate these B and Y terminal fragments. And in theory, if we do one more CAD, these terminal fragments will be fragmented into internal fragments. So in theory, these internal fragments should be coming from subsequent cleavages of terminal fragments. So they should follow the same fragmentation pathways as terminal fragments. So they should follow the very well-known mobile proton model. And by understanding this formation, it can help us develop softwares to aid the assignment of internal fragments with higher confidence and higher accuracy. So how we achieve this goal? How do we demonstrate that, prove that they are essentially following the same fragmentation pathways? So we we took a statistical approach to do this. We fragmented over I think 35 or 40 polypeptides to proteins using top-down mass spectrometry. They range from 1.5 kilodalton to around 10 kilodalton in size. And we generated over 35 or 4,000 fragments, including both terminal and internal fragments. And we summarized their fragmentation propensity in this graphs. So the x-axis here is the 20 amino acid residues. The y-axis is the normalized fragment intensity adjacent to each amino acid residue. So immediately what you can observe here is that we can observe a very similar enhanced cleavages between terminal and internal fragments. So for example, the N-terminal to proline is largely enhanced in both terminal and internal fragments. This is very well known as the proline effect. And also we can see that the C-terminal to aspartic and glutamic acid are both largely enhanced in terminal and internal fragments. So this is known as the acetic acid residue effect. So we can observe these very similar enhanced cleavages between terminal and internal fragments. However, on the other hand, there are some differences that we can see. We can observe a more evenly distributed fragmentation propensity for internal fragments. So what I mean by this is that the N-terminal and C-terminal fragmentation adjacent to a specific residue is much closer in internal fragments. So we can again look at proline. We can see that in terminal fragments, N-terminal to proline is much higher than C-terminal to proline. Is much more significant, this difference. However, in internal fragment, the C-terminal becomes much higher, although they are still it is still very significant, but it is much closer for internal fragments. And this is true also for acetic acid residues and for across all 20 amino acid residues. So this means that we have a more evenly distributed fragmentation propensity. This indicates more energy deposition because we have to deposit more energy to suppress this preferred cleavages that we observed in terminal fragments. So we have more mobile protons available that can bring the fragmentation propensity of a specific residue closer to each other. So because of this, we can prove that internal fragments are coming from subsequent cleavages of terminal fragments, and they should follow a similar fragmentation pathways. So why did we do this? Is that by understanding this formation, we can have higher confidence of assigning some specific internal fragments. For example, if we have a sequence that can match to multiple we have a mass that can match to multiple sequences, multiple possibilities of internal fragments. We have higher confidence of assigning one over another. So this project also contributes to our lab. We developed this software called CLIPS-MS, which is an algorithm for assigning internal fragments in top-down mass spectrometry. This project was led by Dr. Carter Lantz and Dr. Mohammed Znaidi, who were both former LULAB members. And excuse me, I quite enjoyed working with them together on this project. This CLIPS-MS, it can assign both terminal and internal fragments in top-down mass spectrometry, and it does so by generating a theoretical terminal and internal fragments and compares them against observed deconvoluted mass list. So I encourage you to check out this publication if you are interested in using this program, and it has also a step-by-step. It has a link to it has a step-by-step guide to how you can use this program if you want to analyze your data to include in internal fragment analysis. So all the data that I have shown and will show in this presentation will be using this program. So after doing this study, after understanding the fundamentals, obviously we wanted to get into applications. So the first application actually we did was to try to apply internal fragments for disulfide intact proteins. But for the interest of time, I'll skip that specific project, but focus more on the biotherapeutic protein characterization. So we for these very heterogeneous biotherapeutic proteins like monoclonal antibodies and antibody-drug conjugates, in industry, they are routinely analyzed by the bottom-up approach. Reduced peptide mapping is one core assay we do right now every day. However, like during my graduate school time, we were very interested in top-down characterization of these very heterogeneous molecules. We know that it's going to be challenging, but we wanted to see if using internal fragments, can we actually get better results to achieve a better characterization results. So just a very brief introduction on these heterogeneous molecules. They are very large. Monoclonal antibodies, they are about 150 kilodalton in size. They consist of two different regions, the Fab region, antigen binding region, which is responsible for antigen binding, and the constant region. And the constant region also has a very complicated glycan profile, and this whole molecule also has a number of disulfide bond linkages. So it is very heterogeneous. That's why it's very challenging to characterize these molecules by top-down mass spectrometry. So our lab was very interested in using top-down to characterize these molecules. This is a general workflow of this project, of the top-down project. The experiments are actually pretty simple. We just dissolve our protein of interest, the mAb and ADC that we're interested in ammonium acetate, and then we do direct infusion of them into our UHMR, which is also modified to add a EXT cell to do ECD fragmentation. We can isolate each charge state and then do ECD fragmentation to generate these very complicated MS2 spectrum. So as you can see, the experiments are actually pretty simple. What's tricky here is data analysis. We have to think about how we can separate the analysis light chain and heavy chain in the same spectrum, because we're doing top down. So they actually do both exist in the same spectrum. We also always validate our internal fragments afterwards. So it took us a while to figure out the best way to do data analysis. Here I'll jump directly into the data. So the first thing we're interested in is obviously sequence coverage. We know that sequence coverage is a large limitation of top-down mass spectrometry, especially for large proteins. So in this sequence coverage map, this is the heavy chain of NIST maps. The orange part, the orange residues are the complementarity region or CDR region, which is very important for a map. It is responsible for antigen binding, for potency. And these gray shaded areas are the disulfide constrained region. And these horizontal lines above the sequence shows the terminal fragment sequence coverage, and these lines beneath the sequence shows the sequence coverage combining both terminal and internal fragments. So if we only consider terminal fragments, we can achieve 27% sequence coverage of the heavy chain. However, if we also consider internal fragments, that is increased significantly to 69%. And this significant improvement is largely due to the ability of internal fragments to access the interior, the middle part of the sequence region that's constrained by multiple disulfide bonds. For example, if you look at here and here, they are kind of exclusively accessed by internal fragments. So this table summarizes the sequence coverage of the whole mAb molecule. So the bottom line, the takeaway message here is that we can achieve 76% after combining both terminal and internal fragments for the entire mAb molecule. So for the 150 kilodalton molecule. So this is, I think, the by far the highest value I've seen from a top-down experiment on a mAb molecule. And this is largely due to the ability, as I mentioned before, of internal fragments to access the disulfide bonded regions. But however, there's also limitations here, especially in the CDR sequence. Obviously, this is the very important sequence. We wanted to access unambiguously to determine their sequence coverage 100%. However, we can only achieve a 60% sequence coverage. So there is still a lot of room for improvement here. So this is for sequence coverage. The next thing we wanted to look at is post-translation modification. Specifically in this case, is the disulfide bond connectivity, because these mAb molecules have a large number of disulfide bonds, and we have to determine their connectivity unambiguously. And these disulfide bonds are very important critical quality attributes in monoclonal antibodies. So what we did was to use internal fragments, and actually just fragments in general that retain intact disulfide bonds to determine their connectivity. So in this example, this is a sequence. This is a protein sequence with 2 intact disulfide bonds. So 4 cysteines here. Our goal, so what we did is to apply a hydrogen loss on every single cysteine in our data analysis to indicate the integrity of these disulfide bonds. So our goal is to generate as many as possible of these green fragments that retain the intact disulfide bonds. So they traverse intact disulfide bonds. So these two traverse one, and these big green fragments traverse both intact disulfide bonds. And we wanted to avoid or limit the formation of these orange fragments. These orange fragments cleave these disulfide bonds and may lead to mismatched dehydrocysteines. For example, these two cysteines are not connected to each other, but they are crossed by these orange fragments. So in order to generate these orange fragments, obviously we have to cleave the disulfide bond, and that is something we wanted to avoid. So that's why we chose to only use HCD, which is known to maintain the integrity of disulfide bond to achieve this goal in this specific analysis. So this is an example to show the heavy chain of the NIST map, the results of heavy chain. In this fragment location map, the x-axis is the amino acid number. Each horizontal line represents a specific fragment that is generated. So as you can see, we have generated a lot of these green fragments that traverse intact disulfide bonds. But at the same time, there are also a few cases that we generated some orange fragments that leads to mismatched dehydrocysteines. So for the disulfide bonds 1 and 4 that are close to either termini, they are traversed by 89 terminal and 9 internal fragments. But for the middle 2 disulfide bonds, they can only be traversed or determined by internal fragments. There are also some fragments that traverse or determine 2 disulfide bonds. There are 13 terminal and 12 internal. But the middle 2 disulfide bonds can only be determined by internal fragments. So as I mentioned, we generated 6 orange fragments that leads to mismatched dehydrocysteines, but compared to the large volume of these green fragments, I hope that I have shown you that we can actually use these fragments, both terminal and internal, to that retain intact disulfide bonds to determine their connectivities. So this is for PTM determination. So We also used this workflow to characterize an intact ADC, antibody-drug conjugate, to determine the drug conjugation sites. But I'll leave that part to the next project. So after doing the previous project using top-down mass spectrometry, obviously you can see there's still room for improvement. Like, we didn't achieve 100% sequence coverage, for example. We cannot unambiguously determine the CDR region sequence. And for ADC characterization, which I skipped, there's still the drug conjugation site determination coverage is still not ideal. It's far from ideal compared to bottom-up reduced peptide mapping. So we wanted to improve upon our top-down characterization. So that's why we chose to do middle-down mass spectrometry. So in middle-down, we do a reduction of the molecule into subunits, right? So instead of characterizing a 150 kilodalton protein, we're now characterizing a 25 kilodalton protein, which should be much more manageable and should theoretically give us more improved characterization results. So in this project, this is a workflow of this project. So the first step is a sample preparation that we need to reduce the intact mAb and ADC using IDES fabricator and also disulfide reduction to break them down into 3 different subunits: LC, FD, and FC subunits. And then after that, we did a direct infusion of these 3 subunits into our UHMR. The reason that we chose to do direct infusion instead of LCMS, traditional LCMS, is that the fragmentation method we use here is ECD, and this ECD on our UHMR, it's kind of tricky to tune. It has so it requires a lot of tuning, and every time we change a parameter, if we do LCMS, every time we change a parameter, that is gonna take a full chromatography like runtime. So like 15-20 minutes, something like that. So it is kind of time consuming. That's one reason. Another reason is that, again, this ECD on our instrument it's kind of tricky to do, and it requires a lot of averaging to get a good quality data with high signal to noise ratio. So if we are to do LCMS, the time that we can average our data is kind of dependent on the peak width of our chromatography. So that's why another reason we chose to do direct infusion so that we can average a long time. But at the end, we were still able to separate these subunits on the M/Z space, and then we can isolate each subunit and do ECD fragmentation. Again, the tricky part is data analysis, but thanks to our experience from the last project, we were able to figure this out much more quickly this time. So as I mentioned, we always do validation of all our assigned internal fragments, and then we can combine all the each isolated charge state for each subunit. So this first again, we are interested on sequence coverage, right? And as I mentioned before, we can separate each subunit on the M/Z space, and we then do a ECD fragmentation. We always do this kind of validation of all the internal fragments to match their isotope profiles to make sure that they are real ions. And this graph shows the sequence coverage results of all 3 subunits. The top panel is terminal fragments. The bottom panel is internal fragments. So as we can see, terminal fragments, we're still missing some part of the sequence. We have some gaps here and here, especially in the middle part of the sequence. And this can largely be complemented by internal fragments. If we look specifically at one subunit, in this case, we can look at the FD subunit. this is the same fragment this is the same sequence coverage map that I showed before. So if we only look at terminal fragments, we can achieve a 72% sequence coverage, which, you know, is not bad considering this is a 25 kilodalton protein. And but that is because we only have 54% sequence coverage in the middle part of the sequence. And for the CDR sequence, we can achieve 94%, which is pretty good. We're only missing 2 cleavages here. I think one here at the beginning of CDR3 and one here in the middle of CDR3. However, if we also add internal fragments into the picture, that full sequence coverage is jumped from 72% to 97%, so almost 100%. And this is largely due to the middle part of the sequence, so 54 to 95%. And we can also have one more cleavage in the CDR sequence. So here it is now covered, and we're only missing this first cleavage here. And this cleavage can actually be complemented by this residue in the CDR sequence can be covered by this subsequent cleavage. So this table summarizes the sequence coverage results of the entire map. So essentially, we can achieve almost 100% sequence coverage for all 3 subunits, 98, 97, and 97. And that is, again, largely due to we can access now the middle part of the sequence. And more importantly is that we can unambiguously determine the CDR sequence this time. So the only one site that we're missing is the one that I showed in the previous slide, and which can actually be complemented by the subsequent cleavage on the same site. So this is for sequence coverage. We can see there's a significant improvement. And as I mentioned before, we also used this same workflow to characterize an ADC, antibody-drug-conjugate. So this ADC we characterize here is a lysine-linked ADC provided by our collaborator from Amgen. So essentially, every single lysine can potentially be conjugated. So we wanted to determine the drug conjugation site and to cover as many as possible conjugation site. So in this ADC, we have to do two separate reduction experiments. So one with both IDES fabricator and TCEP, one with only IDES. The reason is that the FD subunit is very close in mass to the FC subunit. But at the end, we can actually observe all 3 subunits conjugated to the DM1 drug payload. So the next step is obviously isolate each drug conjugated DM1 conjugated subunit and then do ECD fragmentation. We then obviously always do this validation on the naked internal fragments, drug conjugated and naked terminal fragments. The same thing for FC subunit, ECD fragmentation, and we can validate these drug conjugated terminal and internal fragments. This is a fragment location map that I showed before. This is for the LC chain. The x-axis, again, is the amino acid number. Each horizontal line this time represent a specific fragment that is conjugated to the DM1 drug payload. And there are 11 potential conjugation sites on the light chain on the LC subunit. They are shown here as the black dash lines. So there are 11 lysine residues. We generated 60 DM1 bond terminal fragments and 85 DM1 bond internal fragments. So if we only consider terminal fragments, we can only localize one conjugation site and identify one additional site. So what I mean by identifying here is that it cannot be pinpointed to a specific site, but can only be narrowed down to a few sites. So However, if we also consider internal fragments, we can localize 7 conjugation sites and identify 2 additional sites. So that is 9 out of 11 conjugation sites on the light chain that is determined. So in this table, we summarized our drug conjugation sites results. So the takeaway message here is that out of the 90 potential conjugation sites of the entire ADC, we can determine 72 of them. So that's 80% coverage, which is comparable to the 83% achieved by our collaborator using bottom-up reduced peptide mapping methods. So in summary, for this project, I hope I have shown you that with the help of internal fragments, by using a middle down approach, we can achieve a comparable characterization results to reduce peptide mapping. So in conclusion, for my talk today, I have shown 3 different projects from fundamentals that CAD generated internal fragments. They follow similar fragmentation pathways to terminal fragments, and they should be able to be explained by the mobile proton model, which can help us develop softwares that can assign internal fragments with higher confidence and more higher accuracy. And from the application point of view, internal fragments can improve the both top-down and middle-down mass spectrometry characterization of biotherapeutic proteins. So last but not least, there's a lot of people to acknowledge and to thank. My former lab, the whole entire Lu lab members, my advisors, Dr. Joseph Lu and Dr. Rachel Lu. They are tremendous advisors and mentors, who have supported me so much during my graduate school research. I can't thank them enough. And Dr. Mohammed Zunaidi and Dr. Carter Lance, shown in this picture. We took this in last year's ASMS. Mohammed flew all the way from Australia to attend last year's ASMS meeting. So they have been tremendous support, not just colleagues, but also friends to get me through the very challenging pandemic time. And all of the Loot Lab members, and this is a picture from our and also our collaborator, Dr. Ian Campazano from Amgen. And he has helped me a lot during the last 2 projects, and all the help that instrumentation help we received from thermal and emission. And I wanted to thank the consortium again for giving me this opportunity to present here, and thank you all for your attention. And I'd like to take any questions. Thank you.
Great, Ben. Thanks a lot for that great presentation. It brings back some memories, even though you just left only just a few months ago. So I encourage people to either raise their hand or put a question in the chat if you have questions for Ben's work. But let me start and ask you. So now you've been at Merck now for like 3-4 months now, and you did a summer internship also at Merck. So you have some perspective on what's needed in the biopharmaceutical world. Can you envision then either a top-down or a middle-down workflow would be the way to go then as far as contributing for drug discovery, especially for therapeutic antibodies?
Yeah, the short answer is like top down. It's still far away, I would say. Middle down, it's still in the characterization field, but there are some work that are being done for middle down. So top down, I haven't seen any, honestly, but there are some work of middle down. I think the more applicable method right now is still just to reduce, I mean, from the intact level, because there's a lot of information that we can get just from the intact level. But in terms of fragmentation, especially the most important attributes that we're using mass spectrometry in industry is PTM quantification, which is still like for top down and middle down, there's still a very long way to go. So yeah, that's my perspective.
Okay, good. Okay, so there's some questions in the chat. Let me just read them to you. Great talk, Ben. Impressive data. Can the electron kinetic energy be used to can the electron kinetic energy be increased to get these internal fragments?
For ECD?
Yes, I'm imagining that. Yeah.
Yeah, the short answer is yes, I think so. So I think we published a paper in our lab a while ago. I think Mohammed was the first author that we used the EID. We compared different fragmentation methods, EID, ECD, CAD, and EID is higher energy, higher electron energy than ECD, right? And I think we did observe more internal fragments in that case.
Yeah, so that could be used. I think if you remember, the study was we were doing native MS at the time. And so I think using higher energy electrons for native MS or native top-down MS really improved the not only coverage, but also you started to see more internal fragment lines.
Yep.
All right, another question. Could you comment, let's see, would the scoring be done differently, or can be extended for observing internal fragment ions in homologous proteins? I'm assuming there will be conflicts in determining internal fragment sequences associated to determine homologous protein coverage.
Uh,
So he's asking about scoring.
yeah. So I think that's a very good point. And like in terms of scoring, obviously we don't have any benchmark or scoring system right now. But I mean, I think this is something that we need to think about in the future, and this is definitely a very important development direction in the future in terms of assigning these internal fragments. Because like you mentioned, there are indeed some conflicts. I do see them a lot in my own data analysis, like one mask can be matched to multiple sequences. What I used is the method that I mentioned in the talk, that the fragmentation propensity, right? Which one is more likely, which site is more likely to be cleaved. So that one will be preferred. But I do agree we should think about a scoring system in the future. But which factors should we take into account is something that we need to discuss, like as a community moving forward.
Yeah. There's a question from Christian Bleiholder that's kind of related. That's why I'm asking. I'll tell you this now. Could you comment on how you handled true, false, positive assignments given the vast number of internal fragments?
Yeah. So what we did was that so first, when we assign the fragments, right, we prioritize terminal fragments. So all the terminal fragments will be assigned first, and the rest of the signals will then be sent for internal fragment assignment. So that's first, and then what we did next is always, after that, we always compare our assigned internal fragments against a theoretical terminal fragment list. So that includes all the terminal fragments with some potential PTMs or neutral losses, for example. And after that, all the rest of the assigned internal fragments we will validate them one by one by the way that I mentioned in the talk, by matching the isotope pattern. So it is a very like manual and also labor intensive and also subjective process. There's not a perfect way to do this right now, and that is the way that we figure out might be the best to avoid, false positives.
Lissa Anderson has her hand raised. Lissa, do you want to turn on your mic or?
Can you hear me?
Yes.
Okay, Ben, the NIST monoclonal antibody was included in the second pilot study of the consortium for top-down proteomics. So there are a lot of people in the community that have NIST monoclonal antibody data by middle down and top down. My question is, are you willing to share your raw data or make your raw data on the NIST monoclonal antibody publicly available?
Yeah, yes.
That's a good idea, Lisa. Yeah, we should do that.
Terrific. I'll email you.
Yeah.
Okay, good.
Thank you.
Let's see, there's one last question, unless anybody else wants to post any more questions. This question is about the EXD cell from emission. Can you comment about the challenges faced with tuning this, for tuning the fragmentation using the emission cell?
Okay, yeah. So it is kind of tricky, especially like when you just get started, when you like first few times using that, because there are 7 parameters from my memory, and they are like initially you don't understand them, like what each parameter means at all, right? And you're kind of just you know, changing values and don't know where to start. So I would say, like, if you want to use it, definitely get a list of parameters. Like they should have a list of like model proteins, or the proteins in this size, you can probably start from here. Proteins in this size, you can probably start from here. So you can start from a specific setting and then go from there. It is much easier that way. So you can, when you just choose one set of parameters for your own molecule, when you can start seeing some fragments, and then you can fine tune from there. This is much easier than, you know, starting from scratch, just building your own, you know, list of parameters. That's my suggestion, but it is tricky.
Yeah, we started publishing the parameters. So not every paper has published the parameters that they use on that EXD cell when they publish a paper using it. And we started doing that actually when you were in the group. Then I think we published some values for the last paper.
Yeah.
And we're writing some papers now where we're going to publish the parameters on the EXD cell as well. But you don't see many standards where you see the parameters right now. It's not like you can, I don't know how many instruments have the EXD cell in the Orbitraps right now. And probably there won't be any in the future now that it's owned by Agilent. But yeah, it's a good place to start if you have some parameters for standard proteins.
Yeah, and I think from what I talked to Carter, they have a EXD cell on their Agilent QTOF, and I think they mentioned that it is pretty similar. It is the same set of 7 parameters from
Yeah.
the thermal UHMR and the Agilent QTOF. So that's probably a good thing that
Yeah.
it is consistent across different platforms.
Let's see, another question just came in. Could you please comment on the sequential fragmentation of terminal fragments to obtain internal fragments, or it's a mixture of both terminal and internal fragments in the generated tandem data?
I'm not sure I understand that
I
question.
think they're asking, are you sure that the internal fragment ions are coming either subsequent to generation of terminal fragment ions, or could they be possibly generated at the same time?
Oh, at the same time, meaning like if so.
Well, yeah, is it sequential? Like make a
Oh, okay.
terminal fragment ion and then you get an internal fragment ion, or could they be happening at the same time? I guess could be one way to ask that question.
Yeah, okay. Uh, yeah, so it could be the same time. I mean, the subsequent, it's not exactly like a time course thing. It's more um like a process thing. I'm not sure if that makes sense though. But it also depends on the instrument you are using, right? How long the fragments are spending in the collision cell. Like if you're having a orbitrap, it's totally different when you are doing a CAD on a QTOF or on a FTICR, let's say. The trapping time of your fragments are very important. So I think it is possible that, so I think both cases are possible. So it can be like internal fragments are after, or they are generated at the same time. What I was trying to say in the talk is that this internal fragments from a process point of view, they should be coming from subsequent cleavages of terminal fragments, if that makes sense.
Yeah, actually, when you gave that part of the talk, I was thinking, we never did the study on ECD generated internal fragments. So your original study was CAD generated internal fragments, and in principle, someone should do the ECD version of that same study.
Yeah, it might be interesting to look at that.
Okay, so last question then. Paul Danis just asked, do you have a link where people can get the Clips MS software? So how do people get access to Clips?
You can access it by, let me see, going to this publication here, this publication here.
Yeah.
And then there's a link in the publication that will link to the software itself.
Yeah, I think the easier way, though, I mean, I'm happy for people to read our paper, but the easier way would be to
It's
go to
to just Google Clips MS, I
right.
guess.
Well, yeah, just go to GitHub
Yeah.
and then find the Clips MS software. So
yeah, I guess, yeah, I should mention that there's a link in this paper to the GitHub repository of our of Clips, and it has the step-by-step guide and how you should build your modification file and everything like that.
right. So someone just posted the GitHub link
Yeah,
there.
there we go.
Yeah, and I think that hub will take you to the latest version of Clips, which is the version 2.0 or 2.1 version, I think. I don't know. Carter is on the line, so he can he can answer that. But yeah, the program is freely available. It's on GitHub. Okay, so why don't we end it today? Thanks everybody for joining us for this Thursday seminar by Ben Wei. I hope to see everybody at the ASMS conference. Remember, we have a top-down proteomics evening workshop on the topic. There's also an oral session on top-down proteomics. So I hope to see everybody in Anaheim. It should be crowded. Go to the meeting first before you go to Disneyland. That's what my recommendation is. Other than that, thanks everybody for joining us, and we'll see you at next Thursday symposium. So bye, everybody. Thanks, Ben.